Now we'll begin with glycolysis, but first a walk through the basic parts of cellular respiration. Cellular respiration is broken up into several steps, and this slide here is one that you should really come back to later to review what you've learned. Glycolysis is the first step. It takes place in the cytosol, meaning outside the mitochondria in the liquid of the cell. ATP is made through substrate level phosphorylation. It's going to make a product known as pyruvate, which we'll talk about in a moment, which goes into the mitochondria, where it's changed slightly and then goes into the citric acid cycle. And eventually, parts from each of these steps end up in this process known as oxidative phosphorylation, which takes place at the electron transport chain. And that's where ATP is made through oxidative phosphorylation. A lot of lingo here. It'll take a lot of review and a lot of studying to make sure you get it. Let's break down glycolysis. Summary. It takes place in the cytoplasm, or I usually say cytosol because that's more accurate. That's the liquid of the cell. Okay. The cytoplasm of the cell, the cytosol, the liquid of the cell, meaning this is a very ancient process that probably evolved before organelles, because bacteria do this in their cytosol. Eukaryotes also do this in their cytosol. Now, we're going to follow one molecule of glucose in this lecture, okay? One molecule of glucose, C6H12O6, okay? So you ate a sandwich, or let's say a Pop-Tart. It had sugar in it, right? You digested that sugar in your stomach and intestines. That sugar got into your blood. That blood took that sugar to a cell in your brain or your big toe, and that piece of sugar, that little molecule of sugar, is now inside of your cell, and that's where we're starting here. So digestion is over. We're now at the cellular level. That piece of sugar, that molecule of sugar, is inside your cell. First thing that's going to happen, your cell's going to burn 2 ATPs to get the reaction started. Basically, like the activation energy needed to get this molecule to break apart. Overall, what's going to happen? 2 ATPs are going to be consumed. 4 are going to be produced. So we get a net gain of 2. So we get a net gain of 2 because we burn 2, gain 4. 2 NADHs are produced. Those are those molecules that carry electrons. 2 of those are going to be made. 2 molecules of pyruvate are going to be produced. So let me just show you something. I usually draw glucose as a stop sign. That's terrible, but you get the idea. I'm going to draw it as a chain of carbons for now. So pretend the stop sign just opened up and all the carbons are stuck together. There's 1, 2, 3, 4, 5, 6 carbons, and then hydrogens attached to it in a glucose. Basically what happens in this reaction is several steps. You can look in your book. There's a bunch of steps. There's a lot of little things that happen, including this thing becomes glucose 6-phosphate for a while, a molecule we talked about in a previous lecture. But ultimately, 2 pyruvate are produced, and a pyruvate is a molecule composed of 3 carbons. So basically what we've done is broken this chain in half. And these are now called pyruvate. And there's 2 of them, of course. Here's a diagram showing some of this. There's glucose. It comes into the cell. 2 ATPs are used. And this is called the energy investment phase. This is the first step. Then, as the process continues, we're going to make 2 NADHs. We're going to get 4 ATP produced, and pyruvate is produced, right? And then down at the bottom, you see kind of the net results of this. We get 2 pyruvates produced. We get water produced. I'm not as worried about the water. We're going to focus on the major inputs and outputs here. So 2 pyruvate are produced. 2 ATP are produced. That's net ATP produced. By net, I mean we used 2, so we get 2. We're 2 ahead at the end of this. And 2 NADHs are produced. Another thing that is produced that I won't stress right now, but I do want to point out because they are very important, are these hydrogen ions that are produced. Those are going to come into play later on. The next step then is the oxidation of pyruvate. Oxidation means to oxidize, to steal electrons, to break apart. So pyruvate, if you recall, is a 3-carbon molecule. So remember, we made 2 pyruvates with the breakdown of glucose. Now we're going to follow one of those pyruvates for a little bit. This, in prokaryotes, this takes place at the plasma membrane. So we have enzymes stuck in the plasma membrane. But in cells like ours, in eukaryotes, this takes place in the mitochondria. So this enters the mitochondria. This pyruvate comes into the mitochondria. And what's going to happen is we're going to oxidize the carboxyl group. You might not know what that means, but a carboxyl group is basically a CO2 molecule. It's going to be oxidized from the pyruvate, forming CO2. And we're going to stick a molecule of coenzyme A to this. And so what's going to happen is this is going to be broken down into a 2-carbon molecule. And this carbon right here is going to become CO2. And it's going to leave. You're going to exhale it when you breathe out. That's where some of your CO2 comes from. This molecule here is going to get another molecule stuck to it known as coenzyme A. And that's what acetyl-CoA is, acetyl-coenzyme A. That's the final product of this reaction. So here we can see in this diagram, here's pyruvate in the cytosol, so in the liquid of the cell. This is that carboxyl group. Look at it. It's C and 2 oxygens. It is CO2, right? And so pyruvate comes into the cell through a transport protein. That CO2 is removed. Coenzyme A is attached. And we are going to make another NADH. So you could add NADH as an output here. There is an NADH produced, okay? And we produce acetyl coenzyme A. This molecule right here. Oh, let me step back. So what have we done in this video so far? We took glucose. We broke it in half. We produced a couple of molecules. We produced some electron carriers, that NADH, right? We made some water, we made some hydrogen ions, but we also produced pyruvate, a 3-carbon chain. And in the next step, that 3-carbon chain came into a mitochondrion. It had one of its carbons ripped off, turned into CO2, basically, and that's going to be exhaled as waste. We stole a few more electrons in that process, and we made another NADH. Then we stuck coenzyme A, which is a molecule that's stuck to this 2-carbon molecule that's left over, and it forms acetyl coenzyme A. That coenzyme gives this molecule the right shape to interact with the next set of enzymes in the next video, and that's where we'll start.